Great. All right, so let's get started. So we have a really exciting couple of talks here in the Cell Size and Growth series. We have Peter Lenart from the Tobin Lab at the University of Bern, followed by Shayma Nandakumar from the University of Pittsburgh, looking at some interesting questions of the intersection of our interests and developmental biology. All right. So without further ado, let's take down the intro slide. And Peter, if you could share your slides, we can get started.
Thank you very much for this opportunity. This is not the first slide. And I would like to start by saying that even though my project is about size and growth at the level of whole organisms and tissues, it has been inspired by a lot of research from cell size and growth community. So it's a very nice that I can share my research with you here today. So I'm interested in how babies develop into fully functioning adults. And as you all know, babies are very tiny, so they need to grow a lot to become adults. But different parts of their bodies, different tissues, actually grow at very different rate. And because of that, adults are not just bigger babies. We look different, we have different body proportions. For example, in case of humans, babies have much bigger head compared to the rest of their bodies. And this is not just a cosmetic difference. If you saw a person that has too small or too big head for their age, this would be a sign of severe developmental disorder. And so there needs to be a mechanism that can coordinate growth of different tissues with developmental time. So this is what we study using C. elegans as model. Specifically, I focus on C. elegans germline, which is very tiny at H, but very big in adults. At H is composed of only two cells that correspond to around 3% of the body. And then at entry to adulthood, it's composed of around 2,000 cells that actually outnumber somatic cells 2 to 1, and they correspond to 1/3 of the body. So thanks to small size of C. elegans, I can put individual worms into small agarose chambers, and I can array hundreds of such chambers next to each other. And by using live imaging, I can monitor growth of body and germline from H to adulthood in hundreds of individuals in parallel. And thanks to fluorescent markers, I can monitor this growth at very high precision, as I hope you can appreciate from this computationally state in the movies. So from each experiment I do, I can get hundreds of growth trajectories for worms. And individual trajectory looks something like this. And here, only thing I want you to notice is that both body and germline grow quite substantially during development. So using this approach, I have found that germline to body proportions at entry to adulthood are very important because they show a very strong negative correlation with onset of reproduction. So bigger given worm has its germline relative to its body, sooner it will start to reproduce after reaching maturity. So from perspective of individual worms, this leads to sort of resource allocation dilemma, where they need to distribute the limited nutrients they get from food into either growing their soma or germline. So growing germline is, of course, very important because it's needed for their reproduction, but it's actually costly and doesn't benefit growth of the body, while growing the soma helps to make bigger body that can get more resources faster, but doesn't help the reproduction. So there are, of course, many possible ways how you can allocate resources between soma and germline. So let's see what C. elegans actually do. What I observe is that germline growth is multiphasic with with two main phases. First one takes around 25% of the development, and during this phase, germline and body growth are proportional. So germline to body proportions don't increase. But after this phase, germline growth rapidly accelerates, and germline to body proportions start to increase dramatically. So we did some mathematical modeling of this using a very simple model that of resource allocation that assumes Michaelis-Menti kinetics for growth of germline and soma. Basically what we see is that this sort of delayed germline expansion is a very good strategy. And just to give the intuition about this, because I can't go through the math in the time I have. So it makes a sense to delay germline expansion because germline doesn't contribute to growth. So you want to delay its start, but you cannot put it at the very end of development because then it would take too long for germline to catch with the normal proportions. So this seems to be sensible strategy for resource allocation, but it also makes this germline to body proportions very sensitive to variation in size at adulthood. So if we have smaller adults, they are likely also to have smaller germline to body proportions, which can be detrimental for the reproduction. But in C. elegans, there are many ways how to make adults smaller, including many types of common environmental stresses. So I wanted to see if actually making them smaller by these stresses results in these smaller germline to body proportions, or whether they have some mechanism how to adjust for this. Tested several types of stresses. I will talk about two of them to make it simpler. One of them is dietary restriction. For that, I used EAT2 mutant that has impaired pharyngeal pumping, so it eats less food, therefore it's dietary restricted. And the second stress I will mention is osmotic stress, which is just adding salt to the cultivation medium. Following both these stresses, worms grow slower, and they also end up being smaller. But perhaps surprisingly, their germline to body proportions at entry to adulthood are the same as in the control condition. So how do they achieve this? As I told you before, if germline and soma reacted proportionally to these stresses, the smaller worms should also have smaller germline to body proportions. But it is not what we observe. If you think about the scenario, there are kind of two intuitive solutions to this problem. You can either start the germline expansion sooner and reach the same proportion, or you can start it at the same body size, but then you need to accelerate germline growth even more than normally. As it turns out, solution number two is correct. So body size at the onset of germline expansion is the same in all conditions, while this germline acceleration is even faster in stress than normally. This might seem maybe trivial, but actually for me this was very surprising that this body size at this expansion is the same, because these stressed worms grow much slower. So it takes a much longer time to get to this size, and also they end up being smaller. So this size corresponds to higher fraction of their total, like of their final size. And because of that, if I change axis of this kind of illustration to development, it looks very different. So basically, this rate of expansion is delayed in time, while this phase of germline expansion is shorter because, for one, the worms are smaller at the end, and their germline grows even faster relative to body than normally. And I think this is kind of interesting, and from the modeling that we did, it also seems to be kind of sensible thing. And the intuition for that is that you want to delay your germline expansion as much as you can, and under this stress, soma grows slower because it can get nutrition from the environment less effectively. But germline actually gets its kind of resources from soma, not directly from the environment, and so it's to some extent shielded from this detrimental effect. So its maximal possible growth rate decreases less than that of the soma, and therefore it can outgrow soma even more than normally. So it can start accelerating its growth later in development. Okay, so this is how elegans keep their germinal divisions robust in stress. But does this actually help the more important trait, which is onset of reproduction? And it seems it does, because across all these conditions, the onset of reproduction is not changed. So even in this stress, worms take same time to start to reproduce after reaching maturity. And if I plot individual worms on top of each other, this correlation seems to be basically the same in all conditions. So to summarize the first part, germline expansion is delayed to optimize resource allocation. In chronic we see that this delay is even like more pronounced, and that the germline accelerates its growth even more than normally. And as a result of this, this germline expansion phase does not temporarily scale in stress conditions. I will mention one more type of stress, and that's UV damage. And these are used to induce acute stress, because under the microscope, I can just shine UV at worms at a particular time, so it's easy to affect them at a specific time in development. And in this experiment, I always irradiated them with UV after this germline expansion already started. Here, I just want to show you that these worms are really much smaller as adults. And here at this plot, I'm showing body area and germline area and germline to body proportions on y-axis. So here there are 4 conditions. So control, there is no irradiation, and then there are 3 different durations of the irradiation by UV. Before the exposure, all worms kind of grow in the same way, as you can see here. After the UV, control continues to grow normally because it was not irradiated. But if you look at some condition after irradiation, for example, this highest dose, you can see that in this kind of window of time, body basically stops growing, because these dots correspond to the same time deviations. And here, basically, x-axis is the same. But at the same time, germline to body proportions increase, which means the germline continues to grow. And after they reach certain proportions, worms recover and they continue to grow basically at the line parallel with control, which and they reach correct proportions or normal proportions. And this, I think, is quite interesting because it shows that this adjustment basically happens only in this kind of limited interval. And if I plot all the conditions, all basically the same. So in this experiment, I also measured ribosome expression in germline relative to soma. and here using fluorescent marker. And here you can also see that before UV exposure is the same across conditions, but following UV exposure, there is a nice dose-dependent response, which to me suggests that this germline growth adjustment is a really active process. So now I showed you that germline antibody proportions identity to adulthood are very robust to many types of stresses in C. elegans. So I was wondering if we can break this robustness, and if breaking this robustness would actually affect reproductive output. So we managed to do it by two different approaches. I will talk about one of them, and we can break this germline response by disturbing stress signaling. So basically, what we did, we mutated a gene called VHP1, which is a conserved double specificity phosphatase regulating P38 and GNK stress signaling pathways. And here, in wild-type worms, if we add some stress, this germline acceleration is faster. In VHP1 mutant on its own, because it's smaller and its germline correctly adjusts its growth rate, this is even faster, and the proportions there are correct. But if we add any other stress on top of this VHP1 mutant, for example, dietary restriction, it no longer adjusts its growth rate appropriately, and we end up with smaller worms that have smaller germline body proportions. Accordingly, these smaller worms also have significantly delayed onset of reproduction. And again, if we plot this at individual level, you can see that regardless of this correlation between germline body proportions identity to adulthood and onset of reproduction is almost perfectly preserved, which to me suggests that this is, very useful correlation at least. So, in summary, what I showed you today is that C. elegans delays its germline expansion to optimize its resource allocation between soma and germline. In stress, this delay is further expanded, and germline goes even faster. So this doesn't scale temporally. And if we break this acceleration, then we get smaller worms that have smaller germline to body proportions and also have delayed reproduction. Okay, so these are all data I measured myself, and I know that everybody loves C. elegans, but for last few weeks, I'm thinking that perhaps this phenomenon I see here might also occur in other animals. And I think this because I stumbled across this study that studied malnourished children in Pakistan, and they reported that malnourished kids have delayed onset of puberty, which is well known for many, many years. But what they also mention is that puberty completion in these kids is not significantly delayed, which to me seems very similar to what I observe in my worms. And also, as you likely all know, puberty starts earlier than it used to start. But start of puberty actually changed much more than the end of puberty, which again, to me, resembles very much this dynamic that I see in C. elegans. And of course, perhaps this is just coincidence, hard to say at this moment, but also perhaps there is kind of conserved, this conserved mechanism of germline maturation across different species. With that, I would like to thank to everybody from our lab for making it a nice place to work, and also especially to Ben for having me. Also, I would like to thank to Swiss National Science Foundation for funding my research, and University of Bern. Thank you very much for your attention, and I'm looking forward to any question.
Great. So, as always, please put your questions into the chat and then we'll get to them in turn. So, I'll start off with kind of a definitional question. Like, what is the end of human puberty mean? So, like in the data that you
Uh,
showed at the end, that was kind of provocative, but like, I don't
so,
know. Like, you can either
so,
reproduce or you can't, but what are the other?
so end of puberty in humans, typically in female. I, Actually, there is more way how to measure this, but typically at females, you would consider end of puberty to be menarche, which is the first menstruation. That's one criterion. Another thing, you can look at its so-called Tanner scale, which looks at the maturation of at maturation of sexual organs, for example, breasts or other reproductive organs, and that's slightly different. So, yes, for males, it's a maturation of
But
testicles.
then the onset is a bit
The onset is based, so the one I was referring to, it's based on breast maturation, and it's like called B2 stage. I'm not really expert in this, but it's basically when the nipples start to kind of go, well, start to mature. It's a thing that pediatricians score. Yes.
okay. So we're getting some questions coming in the chat here. Matthew, if you want to unmute and ask your question.
Yeah, thank you for a nice talk. I mean, one thing that, you had this nice kind of genetic way of breaking the stress signaling, right? And uncoupling these things. So if I understand it correctly, the soma and the germline are having different responses in response to stress, but possibly in response to the same upstream signal. So like two different bits of the organism are having kind of divergent responses. Is it understood what might be different about them that allows them to have these opposing responses to the same inputs?
I don't think there is like a clear answer to this. There are several possible speculations. For example, C. elegans is very interesting for, in my opinion, because somatic cells basically don't divide anymore or divide very little after egg is hatched, but germline has a lot of cell division. So this could be one of the reasons that germline actually has more proliferation going on. But we don't know exactly why they react differently, but they react differently.
And then I had one quick follow-up thing, if it's okay, which is I guess throughout all your measurements, you're looking at the germline to body ratio, but how much have you done to match that to fertility? I see that on your last slide here, it does say and reproduction, but does it always match when you mess around with this ratio that it has a linear impact on fertility?
Well, at the onset of reproduction, the whole fertility is a bit more complicated, also just to measure. But there, because I guess if you look at the whole fertility as per se, how many eggs are produced, then it's a bit more complicated. The onset seems to be linear. The correlation, I mean, typically overlaps very nicely. I can move it like up on the scatter plot. In some mutants, for example, mutants in TGF beta signaling, they go much slower, and they are kind of up, but it's the same correlation inside of them. It's just a shifted y-axis. So yeah, I hope that answers your question.
So
Great,
in the
thank you.
end, if I understood, like you end up making roughly the same size egg in the different mutants, like so, or
Uh, I think there are some differences. To be absolutely honest, I haven't measured it like in a great detail, but I think it's known from literature that under some stresses, the eggs are loaded with more vitellogenins and so on. So I would expect there are some differences in size, and we also I think that following dietary restriction, eggs are a bit smaller. So I would expect the eggs there is some variation on that.
Okay. It might be interesting to look at some size mutant worms, right? And see how the ratio is
Yes, I did.
fluctuated.
It's the same.
Yeah. Oh, it's the same. Okay. David, if you want to unmute and ask your question.
So, Peter, very nice talk. So we're talking about the competition between growth and reproduction. I'm wondering whether the nature of the stress determines which you get. I mean, is the worm smart enough to go into reproduction because it wants a genetic solution to the stress that you've provided? Would just making the medium deficient in a nutrient, as opposed to using a genetic mutant, actually promote soma investments? Because I gather the bigger the worm, the better the mobility and the bigger chance it might be able to migrate to a richer environment.
So, thank you. If I understood correctly, so the question is if instead of using a genetic mutant to mimic dietary restriction, I make a mutant, I just restrict the food, if it reacts differently. I actually, this would be very hard in the setup I have, because if you want to do dietary restriction in worms, you need to do it in liquid medium, which is very hard to measure the worms the way I do. I suppose it's possible, but for example, after this UV radiation, I see that the germ antibody proportions are conserved, but most of the worms actually don't reproduce because I guess the UV is really messing up with the meiosis and with the germline. It's possible. I haven't tested this particular case, not because I haven't thought about it, but it was kind of technically hard to do.
All right. Mimi, do you want to ask your question? Or do you feel I was covered by Matthew?
I think it was pretty well covered, so thank you.
Okay. All right, then we'll move to Alex.
Hi, that was really cool. I was curious about like if you have tried ablating one of the germlines and if the other germline can compensate its growth, and if you think that through some other experiment that there's some mechanism it can sense the total amount of germline it has.
Thank you. So I haven't had ablating one of the germline arms, but I try to use genetic mutant that at 25 doesn't grow germline at all. So GLIP1 deletion. Point mutation, sorry. And these worms that don't grow germline actually develop faster, around 2 hours faster. So not like something you could see on a plate, but they develop significantly faster. So from that, I know at least that there is some like a growth cost for growing germline. It's never so clear with the mutant, but that's what I assume. And ablating one germline arm would be indeed very nice experiment, but I haven't done it to date.
All right. So I think with that, let's thank Peter for such a fun talk and really, really interesting stuff. And now we're going to move to Shayma. Shayma, I see your slides already up, so whenever you're ready, let's get started.
Great. Thank you. Good morning or good afternoon, good evening, everyone. I'm quite happy to be presenting at this seminar series, which I've watched and enjoyed over the years. So happy to be here. I'm a postdoc in the Vasudevan lab or the fly stress lab at the University of Pittsburgh. And my work in the lab here has focused on addressing two key questions. The first of which I'm going to talk to you about today, which is how do different cells or various cells within a tissue coordinate a stress response? So, something that we think about a lot in the fly stress lab is how highly metabolic tissues, such as the liver, adipose, or the pancreas, all of which share high biosynthetic burdens, endure constitutive stress, such as ER stress or the unfolded protein response, as a burden of their function. So they have to endure stress while performing their function. In fact, it's one of their hallmark features. And as our lab's name probably suggests, we rely quite heavily on the small but mighty Drosophila melanogaster to address some of these questions. And we primarily use the fat body or the adipose tissue of Drosophila to study how cells respond to stress. So, for those of you who don't think about the Drosophila fat body every day, let me introduce this tissue to you. Much like the C. elegans germline, the Drosophila fat body in the larval stages occupies a large proportion of the animal's body. And it's considered both liver and adipose-like, and it's the tissue that both undergoes extremely rapid growth and also facilitates the exponential growth that Drosophila larvae see from egg to pupa. And the animal grows about 200-fold in less than 5 days, and most of it is supported by this tissue. So the fat body has very important endocrine, secretory, innate immune, and nutrient storage functions. And ablation of this tissue results in complete lethality of the animal. It's also an extremely easy tissue to dissect, so it's quite fantastic to work with, right? So one of the reasons I was recruited to this lab was to study the heterogeneity in stress responses in the fat body. And so some of my early studies interrogating this using a cell stress reporter, details of which are here, revealed an interesting pattern. Well, I was initially recruited to study how some cells show high levels of stress and some cells show low levels of stress. But what I noticed almost immediately upon joining the lab is that whenever cells show disparate levels of stress reporter expression, they seem to appear in pairs, which I hope you can see my mouse. You can see pairs of cells showing high or low reporter expression. And so this sort of reminded me of something that I'd seen before. And before verifying that, I went into the literature and saw that clonal mosaic clonal expression experiments over the years across decades from multiple labs also see what we've seen, but haven't really made a note of it, which is that whenever you make mosaic mutants or transgenic overexpression, you see a pattern where 2 cells or 4 cells in clusters show transgene expression. And this reminded me of something I'd seen in other somatic tissues in Drosophila that possess ring canals. And so, what are ring canals? They're known by different names across the animal and plant kingdoms. And while they might be structurally different, they are generally result of incomplete cytokinesis that results in cells sharing some or most of their cytoplasm. In Drosophila, they're quite well studied and known as ring canals. And in the germline, their function is quite well known. And multiple cells in the germline are connected to one another through ring canals formed by incomplete cytokinesis. And the function is to ensure maternal loading of proteins and mRNAs and even lipids into the oocyte. And this happens both in male and female germline. They're present also in mammals and perform quite the same function. In yeast, septin rings perform this the function of coordinated growth between daughter cells. And while they're structurally very different, plasmodesmata in plants are also a result of incomplete cytokinesis and allow for nutrient distribution across multiple cells. And so we're not quite sure we didn't quite know what the function of ring canals may be in somatic tissue and whether they're even present in the fat body. So that became sort of my next question. So long story short, or not very long story, is that the fat body does indeed have ring canals. We can look for them by using a transgenic reporter line that marks Pavarotti GFP, which is Drosophila equivalent of KIF23, which is frequently found at sites of incomplete cytokinesis. And sure enough, we find ring canals or ring canal-like punctate throughout the Drosophila fat body in a tissue geometry where one adipocyte is paired to exactly one of its neighboring cells through this Pavarotti GFP puncta. However, the Drosophila fat body is large tissue with extremely large cells, but these puncta are extremely small. In fact, they're nanoscopic. And we had to go in and do super resolution microscopy followed by deconvolution to even determine that they're open. And this is an image showing confocal micrograph. This is deconvolution post super resolution microscopy. And the internal diameter of these ring canals are about 260 nanometers when the cells themselves can be a couple of orders of magnitude larger. And so one very interesting thing that I found is that while the animal and the fat body grows quite exponentially through development over the course of 3 or 4 days, the ring canals themselves do not grow at all. In fact, neither the inner nor the outer diameter, which measured here, shows any change despite exponential increase in nucleus size and cell size. This is also a result of polyploidization. So the Drosophila fat body at the last larval instar stage comprises on average about 250 copies of the genome per nucleus. So what we think is happening is that these ring canals form at the end of the last mitotic cell cycle, pairing sister cells, and then individual nuclei undergo about 7 rounds of endoreplication, giving rise to paired adipocytes, which have 250 copies of the genome. This is quite similar to something that happens in the Drosophila prostate, where cells undergoing complete cytokinesis, but then also binucleate. So that's a very interesting, different cell cycle modality. So check this paper out if you're interested. So another interesting thing that happens that we know very little about is that during metamorphosis in the pupa of Drosophila, so after the larval stages are complete, while the adult is incubating, the adipose tissue seems to individualize and take on like a non-professional phagocytic role. And very little is known about this, but it's something I'm excited to explore in the future. Okay, so now that we've found that adipocytes are potentially paired to one another through ring canal-like puncta, my next question became, are they functional? Do they share a cytoplasm? These are very, very small openings, and the cells are huge. So is the ring canal functional? And so to address this, we designed a fairly simple photobleaching after recovery experiment where fluorescence recovery after photobleaching experiment where I simply dissect fat bodies and embed them in a low melting agarose dish and photobleach individual cells to see the recovery of proteins that may traverse ring canals. So I'm just going to show you an example of this. So in this video, I'm going to bleach this cell. And I know that this is the sister cell based on ring canal reporter that I'm not showing in this particular video. And here's a neighbor that it's touching, but not connected to through a ring canal. So the video is not playing. Let's see.
I think you might have to switch your pointer.
Yeah, thank you. Okay, let's see. Okay, all right. Thank you. Okay, so this is the cell that's being bleached. This is the sister cell, and this is a cell that it's a neighboring cell that's not connected through a ring canal. So what we see is a pretty rapid recovery of photobleaching. So the cell that's bleached drops in fluorescence and rebounds quite quickly. The neighboring cell that it's connected to, the sister, shows a concomitant increase in fluorescence. And the cell that it's not a sister cell shows almost no change in fluorescence. So I'm just going to play that video again so you can see it quite quick. As a control, if I bleach both sister cells, we see almost we see absolutely no recovery in fluorescence recovery after photobleaching. So using this assay, which is just cytoplasmic GFP, as a proof of principle to show that some proteins can traverse these ring canals, I sort of determined that this time frame is sufficient to only measure what is going through the ring canals. It's too short of a time frame for any new GFP synthesis. So we proceeded to ask what other cargoes can pass through the ring canal. And I hope I have enough time. Right. So using the beauty of Drosophila genetics, we've done this with various organelle-bound fluorescent proteins. Here's an example with ER-bound GFP. So this is endoplasmic reticulum GFP. Just bleach, recovery really quickly. So what we see is roughly 30 to 40% recovery within a matter of 3 to 4 minutes. And that's quite remarkable because these ring canals are less than a micron large, and perfect recovery would be 50% because the neighboring sister cell is sharing roughly half of its cytoplasmic content. So I thought that this recovery is quite impressive. We also see that Golgi bound proteins travel through ring canals just as rapidly, if not faster, than ER. And I'm not going to show you this because I don't think I'll have time, but we don't see any mitochondria going through the ring canal. So there seems to be a very specific set of cargo that ring canals allow transport of. And we also think that lipid droplets are too large to go through them. And if they do, we probably don't have the right resolution to study that. Okay, so next question became, can we disrupt these ring canals? So I'm showing you an example of a mutant of Kelch, which is an E3 ligase that's bound to the cytoskeleton and is required, at least in the Drosophila germline, to maintain the openness of ring canals. And so I did a fat body specific knockdown of Kelch, as well as other septin proteins. Peanut is septin 7, and even a heterozygote of Kelch. So depletion of Kelch does close the ring canals. So the inner diameter of the ring canals becomes much, much smaller. Whereas when I disrupt septin proteins, the ring canal size doesn't change, but the shape changes. And this suggests that all of these proteins are somewhat important to maintain both the size and shape of the ring canal, and that it's probably actively maintained because when I look at third instar larval stage, when the knockdown has been done much earlier in development, we still see that these proteins are required. Okay. So I just want to take a quick detour to point out that I've been thinking about the function of ring canals as it pertains to stress response because that's how my brain is wired. But a very, very recent paper from Anna Franz's lab that came out, I think, last week in JCB has revealed that the fat body has apicobasal polarity. And given what we know about the mid body, which is the structure that is stabilized in ring canals, um, and its role in establishing or maintaining cell polarity, it's possible that the apicobasal polarity that was recently discovered in Drosophila fat body uh, might be regulated by or contributed to by ring canals. So just something to think about. Okay, so going back to what the disruption of ring canals might do to the um, uh, openness of ring canals, its ability to allow intercellular transfer. When I knock down Kelch, the intercellular transfer of fluorescent proteins completely disappears. So in a control fat body, you can see recovery within about 5 minutes, whereas when I knock down Kelch, the fat body is not able to robustly recover fluorescent proteins. So this suggests that making the very small ring canals much smaller renders them not functional, at least for the purposes of intercellular transport. Okay. So my next question became, what are the physiological consequences of disrupting ring canals? As I said, my brain is wired to think about how this affects the stress response of this tissue. And so we turned back to the stress reporter that I showed you in one of my first slides. This is a reporter for the integrated stress response, something that we use a lot in the lab. It's a 40BP transcriptional reporter that responds specifically to ATF4 activity. And so when we knock down Kelch, we see a pretty robust upregulation of this stress reporter, which could be expected because you're knocking down an E3 ubiquitin ligase. But what's more interesting is that in certain outlier cells, we see disparate levels of stress reporter expression between sister cells. And so this graph quantifies the total stress reporter expression. And then when I go in and I measure the difference between sister cells that are paired through ring canals, but the ring canals are now disrupted, you can see that the difference in the stress reporter expression is much higher between sister cells, whereas just cytoplasmic GFP is not. I should remind you that this is a snapshot. So this is not like the cytoplasmic GFP that I showed you in previous slides with fluorescence recovery after photobleaching. These are fixed tissues. Okay, another thing that might be of interest to this audience is that I noticed that the difference in cell size between paired sisters in adipose tissue that has disrupted ring canals is very different. So, and this difference in cell size isn't necessarily a result of difference in ploidy because the nuclei appear to be relatively similar in size, but the cell size is different. So there's a lot of disruption of cytoplasmic streaming and maybe buffering of whatever these cells are meant to share. Okay. All right. So there's increased stress and also increased disparity in stress response between sister cells. Now, what about added stress? We can culture fat bodies for up to 24 hours ex vivo with almost no effect on the health of the fat body. In fact, even when we add tunicamycin, which is a pretty stressful drug, we see that very few cells in control conditions die over the course of 18. This is an 18-hour video. But when we knock down Kelch and disrupt ring canals and add stress to this tissue, the cells die pretty quickly. They really, really don't like it. So what we're thinking is that disruption of ring canals is deleterious for fat bodies when they're subjected to exogenous stress. This is also the case this is not just in the case of tunicamycin stress. I've also treated fat bodies to other stresses. They're famously difficult to kill unless you're messing with multiple pathways. So in this slide, uh, I'm stressing the fat bodies out by adding paraquat, which is an oxidative stressor. And my stress readout is GSTD-GFP, which is a xenobiotic stress response reporter. And so in response to added oxidative stress, impaired ring canals also result in higher levels of stress and also unpairing of sister cells. You can see outlier bright cells that don't have a sister that they could be sharing their stress with. Okay. All right. So in summary, to give you a little bit of details on things that I won't have time to share with you today. What I've shown you today is that larval adipocytes are paired to one another through ring canals, and that they're present throughout the larval fat body. This is not something I had time to show you, but we see it throughout the fat body in both male and female Drosophila. And unlike in the germline, the Drosophila fat body ring canals do not change in size, even though the cells themselves grow. These structures support the transfer of cytoplasmic content, specific cytoplasmic contents. So Golgi, traverse ring canal cytoplasmic proteins do. However, lipids and mitochondria do not. And in addition to all of that, we've done a few physiological assays to show that while ring canals result in higher levels of stress and lower levels of serum proteins, lipid reserves remain unchanged, which makes sense because lipids seem to be compartmentalized within sister cells and not necessarily shared between them. We also see a slight nutrient-dependent developmental delay when we disrupt ring canals, but there is no lethality. So these structures might be adding to the robustness of Drosophila, but not necessarily be required for them. Okay, so everything I've told you today is in press at JCB. There's a lot more in the paper. But until the paper comes out, feel free to read the preprint, which has an earlier version of the story. And before I end, I would like to just talk more generally about how this pairing of multiple nuclei through a cytoplasmic bridge or multiple nuclei sharing some version of the entire cytoplasm or some of it is not a quirk of just the Drosophila fat body. In fact, it's quite common and might be a highly conserved feature of metabolic tissues. So on this slide are just a few examples of binucleate tissues or tissues that undergo binucleation in some way. And the function of it is still not quite well known. There are famous examples here, like the cardiomyocytes and mammary glands that are actively lactating. And another interesting thing I'd like to leave you with is that the adult fat body of Drosophila emerges from a completely unknown set of progenitors, but is also a tissue that has binucleate and tetranucleate cells. And so the need for Drosophila adipocytes to have multiple nuclei seems to be shared despite differences in tissue function across developmental stages. Okay, I think I've gone quite a bit over time, so I would like to end here and say thanks to all of the lab members and fantastic imaging facility, resources, and um, I'm happy to take questions.
Great. Thank you, Shayma, for that talk. So please drop your questions in the chat. I mean, I guess I would wonder, just kind of like philosophically, I guess, like why
Mhm.
do you need, I mean, these are pretty big cells. Presumably they have the different enzymes and whatnot that they
Right.
need. Like why would you I'm still like, I mean, but this is like kind of a general question for
Mm,
polyploid cells. I mean,
right.
in a way, like why would you make a polyploid cell rather than having um, kind of multiple cells make up the same volume in the organism? I think I don't quite understand that. I mean, on the one hand, if you have different cells, you can imagine if there's some damage or something bad happens, you compartmentalize that
Hmm.
away.
Sure.
Whereas here, you're kind of making an argument that the canals help you sort of like share resources to deal with the the stress, right?
Right.
Is that the right way of thinking about it?
Yeah, I think that's how I am thinking about it. You've phrased it similarly to how I think about it. But to answer your first question, maybe.
Hmm.
Um, I think becoming polyploid is faster than making multiple cells. So, in order
Okay.
to give rise to 250 copies of the genome, these cells they're not even undergoing endocycles throughout the 4 days. It's like a matter of, I think, 25 hours. So it's faster to skip G2 and M.
Mhm.
Um, and maybe it's not just volume, maybe it's also surface area to volume ratio. And if you are becoming a polyploid cell, you don't have to increase your surface area as much,
Hmm.
right?
All right. David, would you like to unmute and ask your question?
It's not often that you get to hear about a new cell organelle, so thank you very much, Emma. Um, so I have two questions. A comment about polyploidy. Basically, plant cells do it, and I always
That's right.
thought it was to protect DNA from UV damage.
Yep.
Um, but maybe the enzymes in fat cells and pancreatic cells are also subject to DNA damage. That's
Mhm.
point number one. Point number two is pancreatitis is one of the untreatable diseases still. And I wonder if the few people relatively who drink alcohol or take Ozempic and get pancreatitis are lacking ring canals.
That's an interesting thought. I'm not quite sure if there are ring canals in the pancreas, although I wouldn't be surprised if some cells in the pancreas undergo incomplete cytokinesis. In fact, it's not well acknowledged that in addition to the endocrine pancreas, the exocrine pancreas, one has a function, contributes to the pancreatitis more than the endocrine pancreas, and is comprised largely of binucleate cells. So, potentially, I think that polyploidy might underlie one of the reasons that pancreatic cells are difficult to kill. And so, when they become oncogenic, um, it's it's possible that that contributes to their resilience. Yeah, I hope that addresses your question. Also, with regard to plants, yes, plants do everything better than us. I've always maintained this. They're much smarter than humans and mammals and animals. And one of the primary, I think, one of the ways I think about polyploidy is that having multiple copies of the genome is has been shown by multiple groups, both in Drosophila and other species, that one of the main reasons, in addition to high biosynthesis, is protecting against DNA damage. In fact, Drosophila polyploid cells suppress p53 and p53 regulated DNA damage. So, yes.
Thank you.
All right. Next up, we've got Matthew.
Hi, thanks for a nice talk. So I was wondering if we go back to the beginning of your talk, you kind of said that these canals form just in at the end of the last mitotic cycle. And so what is it that means that they happen then? And what is it that stops them happening in any previous cycles? Is there, and then it also made me think, are there other contexts, other tissues where that's not the case, where you have 4 or 8 or 16 cells all linked up together?
Right. So, I don't know what regulates only the last cell cycle, mitotic cell cycle, to be an incomplete cytokinesis. I think it might just be a time crunch because the mitotic cell cycle happens within 6 hours of these cells being, the progenitors being specified. So it had to be quite rapid. Progenitors giving rise to about 5,000 cells that undergo incomplete cytokinesis. So it might be time limited. Um, Other tissues that show 4 or 8 or 16 linked cells are almost entirely either germline or soma associated with the reproductive organs. So there are cyst cells in the Drosophila testis that are 8 or 16 cells. The follicular epithelium that gives rise to the eggshell of Drosophila, I think, can have up to 16 cells in a syncytium. And I think the syncytial size to function relationship is something we're just beginning to explore.
Cool. Thank you very much.
All right. Laura, did you want to add a comment or just that was just a chat for the group? Okay, we're going to assume that's just some information for the group that endocycles
Okay.
also require lower cyclin CDK thresholds. All right, so with that, let's wrap up this session. So thanks again, Peter and Shayma, for the talks. Really nice. And we'll see you all back here in a couple of weeks. Bye, everybody.